Have you ever wondered if it's possible to inject small robots into your body? Can we inject miniature robots into our bodies to fight diseases? You may be shocked to learn that we are closer than ever to achieving this goal. The field of nanorobotics involves the design and creation of nanoscale programmable molecules that can be used to perform different tasks. Nanobots can be designed to move through the body, and they can be constructed to self-propel, using nanomotors made of bio-nano components, in order to deliver drugs to diseased cells. Nanobots can be controlled through different means, including an external magnetic field, UV light, chemically, and through ultrasound. Using an external magnetic field to propel nanobots is a safe and effective method to control nanobots with magnetic particles. An example of the use of an external magnetic field to control nanobots is the use of a homogeneous static magnetic field and a sinusoidal magnetic field perpendicular to it to move a micro-sized swimmer, which consists of a chain of colloidal magnetic particles linked by DNA and attached to a red blood cell. The chain aligns with the external uniform magnetic field, and the oscillating magnetic field causes it to vibrate. The chain swings and propels the nanorobots along the uniform magnetic field axis. The external fields can be adjusted to control the velocity and direction of motion. Another promising nanobot systems were constructed by researchers at the Max Planck Institute for Intelligent Systems in Stuttgart. 2 propulsion systems for tiny floating bodies were constructed. In one system, the propulsion is generated by bubbles which are made to oscillate by ultrasound. In the other, a current which is generated by the product of an enzymatic reaction propels a nano-swimmer. Urease-coated nanotubes can move in a urea-containing liquid because the enzyme breaks down the urea into ammonia and carbon dioxide. The reaction products generate a current which propels them out of the tube. The nano-swimmers are either thinner at one end than at the other, or the urea is not distributed homogeneously over their surface. This, in turn, results in a thrust. The urease-coated nanotubes are biocompatible, and the products which are generated by the reaction are water-soluble, which means that they won't produce bubbles inside fluids in the body. This system is an example of chemically propelled nanobots. Another example of chemically driven microbots is a system which consists of gold nanoparticle-loaded PEDOT, zinc-based artificial micromotors. The micromotors were tested in mouse models via oral administration. Gold nanoparticles were employed as a cargo due to their widespread use as drug carriers. The micromotors showed excellent acid-driven and self-propulsive properties with high cargo loading capacities. The immersion of the PEDOT, zinc micromotors in the gastric acid results in a spontaneous redox reaction which involves the zinc oxidation and the generation of hydrogen bubbles, which are needed for the propulsion thrust. Another great method for constructing nanobots would be to incorporate the body's own tissues into the nanobot. One example would be cell-like nanorobots that clear toxins and bacteria from the blood. Researchers built these nanorobots by coating gold nanowires with a hybrid of platelet and red blood cell membranes. The work was published in Science Robotics. The hybrid cell membrane coating allowed the nanorobots to perform tasks that are performed by 2 different cells at the same time. First, the task of binding pathogens like MRSA bacteria, an antibiotic-resistant strain of Staphylococcus aureus, is performed by platelet. Second, the task of absorbing and neutralizing the toxins produced by these bacteria is performed by red blood cells. The gold nanowires respond to ultrasound, which gives them the ability to swim around fast without chemical fuel. And the coating protects the nanobots from a process known as biofouling, which is when proteins collect onto the surface of foreign objects, which prevents them from operating normally. The researchers used the nanobots to treat blood samples contaminated with MRSA bacteria. After only 5 minutes, the blood samples had 3 times less toxins and bacteria than the untreated ones. The hybrid coating was created by first separating entire membranes from platelets and red blood cells, then applying high-frequency sound waves to fuse the membranes together. The gold nanowires were then coated with the hybrid membranes using specific surface chemistry to create the final nanobots. Thank you so much for watching the video, and we hope you found it informative.